Hello, I'm Raymond Licorice. I'm going to say a few words about symmetries that knots can have in mathematics. Knots to a mathematician are in a piece of string with no ends, so the ends are joined together, and we often draw pictures of our knots on a blackboard, drawing where the string crosses over itself. Starting from just the one picture of what a knot might be, putting the arrow on in one direction or the other gives me two possible knots, and changing all the crossings and combining with the idea of the two possible arrows gives me two more possible knots. So this is possibly 4 knots. For this 4-crossing knot, there is in fact only one knot, one 4-crossing knot, and it doesn't matter how I put the arrow on or how I change the crossings, I've always got the same knot because I can maneuver one picture to the other one. Over here, however, we have the knot 933, and that really does give 4 different knots.
I'm Joan Berman, and I'm a professor of mathematics at Barnard College and at Columbia University, and we're in my office in the mathematics building at Columbia. What I'd like to tell you a little bit about is braids, and braids are very closely related to knots, and we'll see the connection in a few minutes. So one thing is, you will know one example of a braid, and that's a braid in a person's hair. So here's a picture of it, and of course you can keep going. Well, braids can be used to construct knots or links. For example, I could take this braid, and what I could do is just close up the ends. So there's the braid that we started with, and when you close up the end, what you find is that you have a knot. You might get a link, it might fall apart into some number of components, but in the example I've shown, you get a knot. And here's the braid in a girl's hair, or a person's hair, and it gives a very nice knot when we close it up. This knot is called the figure 8 knot. But of course the question is, if you start out with braids, do you just get a limited class of knots, or do you get them all? And what Alexander showed is that in fact you get them all. This was done in 1923, and I want to show you his proof. So I'll give you an example over here. And his example is again the figure 8 knot. Now it doesn't look like this picture of it, but I'm going to show you that it really is. And if I take this to be my future axis, let's see what the problem is. Well, what we're supposed to be doing is traveling around here, and we're supposed to be doing this so that as you go around, and let me start over here, the polar angle is always increasing. So, in fact, I've got it decreasing, but that's all right. So we're going around this way, and everything seems quite all right, and the polar angle is moving along until you get to this little piece. And this little piece here, the thing has turned around, it's going in the wrong direction. And here you see a picture of the same diagram of the figure 8 knot, only now it's tied to a piece of rope on the floor. Well, you step into this picture, you can step in any place you like, but I'm going to step in right over here. And what I know is that it's this piece of rope that's causing the problem. So Alexander's idea is you just take that little piece, so you know the polar angle function is wrong, and you just take it and you throw it over your head. And now, lo and behold, you pick it up, and what you have is a braid. And there it is, very nice, a closed braid.
So now I'm going to tell you my theorem, which is much harder to prove, and which actually came first. Now you're at a 7th point, and you join it to all the others. Well, I'm not going to bother to do it all. Okay, but, well, maybe I'm going to bother. I've got to join that seventh point to all the six others. Since it's fairly easy, might as well. And now, my clever theorem says that somewhere in there you'll be able to find a knot, a real knotted curve.
This is a Turk's head knot. The whole series of them. You notice how there's three strands. They braid together. They braid in the same pattern as the typical braid here. Goes over. Each strand goes over, under, over, under. So the reason I think it's called a Turk's head knot is that it's vaguely reminiscent of a turbine. Generation of hot air or steam or whatever, it turns around and generates electricity.
Mathematicians would like to classify knots. For example, this is a knot. It has 7 crossings, and one possible way of classifying them would be to describe ways to draw them on the board with certain numbers of crossings. But that turns out not to work very well. So mathematicians, knot theorists, look for sub-collections of knots which are nicely behaved. And one collection is ribbon knots. And you can make ribbon knots by beginning with a few circles, unknotted, unlinked circles. So we'll take 3. And then drawing ribbons to connect them together. So a ribbon is the following object. It's a curve which runs around, and it might have a knot in it. And then you thicken it up so that it actually does look like a ribbon. So that connects the first and third circles together, and then we have to connect the second circle to either the first or the third. So we might do it by running out here, and then all the way around to the third one. And then that's an example of a ribbon knot. The simplest example of a ribbon knot is the stevedore's knot, which is made here in the table out of pop beads. The problem is that when you see a knot before you and it's not drawn in this way, then you don't know where the ribbons are that need to be cut. From long experience, one has discovered that we should cut here and here. And so we'll break the pop beads, and then they should be connected up this way. And then this one is connected this way. And now you find that if you just pick up one copy, it just falls apart like this into 2 circles, which are unlinked.
Well, the theory of knots has found an application to the theory of the Poincare conjecture, which conjectures that every simply connected three-dimensional manifold is homeomorphic to the three-dimensional sphere. So here's the recent application that if one takes a knotted solid torus, in fact, if I can draw one. One takes a knotted solid torus in the shape of a non-trivial knot in the three-dimensional sphere. One removes the knotted solid torus and glues it back in an essentially different way. Then Gordon and Lukey proved in 1988 that the result is never homeomorphic to the three-dimensional sphere. So if one can find a three-manifold made by starting with a non-trivial knot, removing a solid torus neighborhood and gluing it back in an essentially different way, and one can find one that is simply connected, then one will have a counterexample to the three-dimensional Poincare conjecture. So that's quite a challenge that I think is worth somebody taking up.
Donc, je m'appelle François Labourie et quand je suis allé voir mon professeur, Michel Gromov, pour lui demander un sujet de thèse, il m'a proposé le sujet suivant. Il a sorti un bout de papier de sa poche, de sa table, Il m'a dit, combien est-ce qu'on peut faire de nœuds avec un bout de papier? Et donc j'ai travaillé sur le sujet, et la première question c'était, quand on prend un bout de papier trop large, bien sûr, on ne peut pas arriver à le nouer. La seule chose qu'on arrive à obtenir, c'est un cylindre sympathique qui n'est pas un nœud. Si on prend quelque chose de plus fin, par contre, on peut bien sûr arriver à le nouer de cette façon-ci. Mais on a l'impression qu'on peut faire qu'un nombre fini de nœuds avec un bout de papier donné. En fait, on peut construire une infinité de nœuds avec un bout de papier, contrairement à l'intuition. Alors, quelle est la procédure? Bien sûr, on a besoin d'un bout de papier mathématique qui est infiniment fin. Donc, on prend un bout de papier, on le plie ainsi. Ensuite, on le roule. Il ne faut pas être trop maladroit. On obtient ainsi une espèce de petit bâton avec deux appendices comme ça. Si on répète cette opération et qu'on prend un autre petit bout de papier, on les met l'un à côté de l'autre. Et maintenant, comme notre papier est infiniment fin, on peut enrouler ce bout de papier autour de celui-là. Et en fait, on peut faire autant de tours que l'on veut, parce que justement notre papier est infiniment fin. Ensuite, on complète le nœud en ajoutant des petits rubans ici, comme ceci, là, puis un qui va rejoindre cette partie-là à cette partie-là, et tous les nœuds que l'on obtient sont différents.
So lots of people think two knots can cancel. So in other words, that if you have a knot A, and the knot is knot A, and the knot B, and they're tied in the same piece of string, that's knot B, that maybe you can just continuously move that string until it's all unknotted. So let's just imagine that that's the case, and let's suppose that we start off like that at 12 o'clock, and then at 1 o'clock the same picture will look like this, if we're right. In other words, you can just move A and B until they completely cancel. Well, I think anybody who can do that is a magician, because knots don't cancel. And the way I proved this when I was at high school in England is like this. You imagine that you can do it. Let's say the magician can do it. And then, oops, you go out and buy some inner tube. Nice, big, soft inner tube. And you put it around the knot like that, so that what it does, this inner tube, is it swallows A and then follows B. Everyone see that? Let me just get a bit more chalk. So what I'm going to do now is at 1 o'clock I'm going to just look in the vertical plane only. And that vertical plane will contain this yellow piece of string, and it will intersect the inner tube in some loopy manner like this. And there may be places like this that I'm drawing now, where fingers of inner tube went through the plane. Okay. So now I'm going to take my green French chalk and walk along the curve like that, marking it. And it's pretty obvious it's got to come out of top right, because it's not allowed to cross this string here, and it can't get to bottom left or bottom right. So there it is. And now we just ask the magician to undo things, and then we see that what he had was a curve in the inner tube, like this, that goes from top left to top right. So it must be knotted like B. But you can see from this picture that it wasn't knotted at all. So B wasn't knotted, and similarly A wasn't knotted. Two real knots don't cancel. They weren't real knots.
Well, the Jones polynomial was discovered in this office in 1984, and it was a pretty exciting time. Before that, mathematicians had a polynomial invariant of knots. It was called the Alexander polynomial, and it's a polynomial like 3 plus 2t plus 5t squared plus 7t cubed minus 8t to the fourth. Jones came here, he came here to talk to me. He'd written to me and we got together because he had come across representations of the braid group in his work, and he had heard that braids were connected with knots, and he wanted to know if there was any more to it than that. Once we began to understand each other's language, he realized that in fact implicit in his work was a knot invariant, that his class invariants were just required a small modification to make them into knot invariants. So Jones had this invariant, and then the question was, what should we calculate it on? So he said, well, what are some easy examples? Well, the very easiest example you can find is a trefoil. So this was the first example, and the first example we calculated it on, it turned out that it was t plus t squared minus t cubed. Well, there was another polynomial around that's also an invariant of knots. It's the Alexander polynomial. The Alexander polynomial for this knot was known to be t inverse minus 1 plus t. Well, this is Alexander. That's Jones. And so the question was, well, that doesn't say anything. Let's try it on something else. Well, I knew from knowledge from other things that, in fact, these knots, the right trefoil and the left trefoil, are inequivalent knots, but as it happens, their Alexander polynomials are the same. So the next question was, why not calculate the Jones polynomial for the left trefoil? And all this happened in about 5 minutes. Well, it turned out to be t inverse plus t to the minus 2 minus t to the minus 3. That is, each t is just replaced by a t inverse, and of course these are not the same. So by the end of that day, we had calculated a lot of examples, and the polynomial wasn't missing a beat, and it was pretty clear that it was new and that it was correct. And when I went home that day, I thought, I couldn't believe it, that there was another polynomial. It wasn't the Alexander polynomial. It was really different, and it opened up a whole new world of mathematics, and it was very exciting.
Okay, there are a whole series of enzymes. Oh, by the way, I'm Nick Casarelli, and I'm a professor of molecular biology at UC Berkeley. There is a whole series of enzymes that change the structure of DNA. They are, in fact, the major determinants of what makes you different from your mother and father, and that is that you have some process called recombination, or reshuffling of the genes from your parents. Now, how does that occur, the reshuffling? It's actually very simple enzymologically, is that you have two pieces of DNA. This ribbon represents DNA, and they're broken and then rejoined in a particular way. I started with one ring, now I have two rings, and they're linked together. So you've done two things. You have changed the sequence of the DNA, and you've also changed its topology, because now it's a link or a cat name. And the most important technique for studying these forms of DNA is gel electrophoresis. And the reason is that as you coil DNA up, it moves faster in a gel, so that you can actually distinguish something which has 43 crossings in a knot from something which has 44 crossings in a knot very easily by electrophoresis. And this is what you use. It's very simple. All it is a power supply, an ordinary power supply, and there's an electrical potential between these 2 points on the gel, and in between them there'll be something to support the DNA, like gelatin or agar, and the DNA travels through it. And then after a certain point in time, we quit the experiment, stain the DNA with something, and then take a picture of it. These pictures are pictures of knots of DNA that we've taken by an electron microscope. And the one on the left is called a trefoil because it has 3 crossings. And the one on the right is a 4-crossing knot. Often it's not enough for us to say that we make a 13-crossing knot. We have to know which 13-crossing knot is made. And the case, in fact, our record is that we looked at the structure of a 19-crossing knot, and I believe there are about 10 of the 7 different ways of tying a 19-crossing knot, and we predicted the right one.
I'm Sylvia Spangler. I'm in the program in Mathematics and Molecular Biology at UC Berkeley and also in the Human Genome Center at Lawrence Berkeley Laboratory. I study kinetoplast DNA, which is an array of some 7,000 molecules, circles of DNA. It comes in 2 sizes. A very large circle, there are about 10 of those, and all the rest are smaller circles, about 5 to 10% the size of the large circle. This array can be made planar, so that it's basically a tiling pattern that contains both the genetic material and also apparently some structural material. The actual role of the small circles isn't known, but what we're initially trying to discover is how they're actually arrayed. If you take the very large complex and break it apart by some sort of physical means, you can get subunits. Subunits vary in size all the way from the single small circle to arrays of up to 10 molecules that you can actually analyze. What we first look at is what the linkage is between 2 circles. And what we have shown by looking at some 600 molecules that have only 2 circles in them is that it's a singly linked catenane. It's the simplest sort of catenane. However, we have yet to answer the question of what is the number of neighbors that each single circle has. We think it's probably in the range of 3 to 4, but even that doesn't tell us exactly what the array looks like. For example, you could have it be 3, and when you separate it by gel electrophoresis, you would be able to separate something that was 3 in a linear array from something that was 3 as a triangle. Even when we have determined how all of these circles are linked together, one of the big questions will remain, how does it replicate? What happens is this entire array doubles its size, and it is done by each of these circles coming off the array, duplicating itself through a knotted intermediate that we still don't know why it's a knot, recircularizes and reattaches to the entire array to make something that is twice the size of the original array, which is then split down the middle. It happens very, very rapidly. Biologically, the question is how it happens and how it could be arrayed so that it happens rapidly enough to let the cell survive.